day, students! How are you today? Welcome to a new day of more fun and learning. This is Teacher Ariel.
This is Teacher Richessa.
This is Teacher Rose Milin. And this is Teacher Christine.
And this is Teacher Menvi.
And we are the Grade 7 Science Teachers of Mati National Comprehensive High School.
We are here to help you learn science at the comfort of your home.
To start with our lesson for today, I will show you a picture. This is the picture puzzle with clues. Complete the missing word in the box using the picture as a guide. One letter is already provided as a clue. Do you know the answer, class? Yes, you got it right! Congratulations! The missing word is microscope. Now, what do you think would be our topic for today based on the activity that we had? Do you have any idea? Yes, it's correct. It's all about the parts and functions of the microscope. And for today's lesson, this will be the following objectives. At the end of the lesson, students can describe the role of microscope in studying microorganisms. Identify the parts and functions of a compound microscope. List down the steps of using microscope properly. And appreciate the importance of microscope in studying microorganisms. Currently, the world is facing this coronavirus 19 pandemic that took millions of lives worldwide. Many were shocked at how this pandemic spread suddenly and was transmitted from human to human in just a short time. What have caused this pandemic? Yes, you are correct. This is caused by SARS-CoV-2. This is a virus that causes COVID-19 or coronavirus disease 2019. It is highly transmissible and pathogenic coronavirus that emerged in late 2019. How are the scientists able to come up with solutions to stop this outbreak? How can they see coronavirus and study it? What instrument they use to study coronavirus and even other microorganisms that have caused death to millions of people? Yes, with the help of the instrument called the microscope. What is a microscope? It comes from the two words micro and scope. Micro means too small to see, and scope means to look at. It is used to enlarge and magnify an object's image which cannot be seen by our naked eye. And a Dutch father and son spectacle maker named Hans and Zacharias Janssen invented the first so-called compound microscope in the late 16th century. What is the role of the microscope in studying microorganisms? Scientists use microscope to study the physical and internal characteristics of microorganisms like coronavirus and others. It unveils the hidden characteristics of microorganisms, its structures, and chemical compositions that cannot be seen by the human naked eye. Now, who among you here have already seen the microscope? Who among you here have already used the microscope? We are here to help you to learn more about the microscope. Let us first identify the parts and functions of the microscope.
And today we are going to discuss more about the microscopes. Microscopes are of different kinds. The most common microscope used in school laboratories is the compound microscope. Here are its parts and corresponding functions. The first part of the compound microscope is the eyelid. eyepiece is also known as the ocular. It also contains the first lens and it is located on the top of the body tube. The next part is the body tube. It is a long tube that connects to the objective lens and attached to the revolving nose piece and eyepiece. The next part is the objective lenses. It is used in combination with the eyepiece to provide a range of magnification. And it is located on the nose piece at the bottom of the body. The next part is the revolving nose piece. The revolving nose piece is located at the bottom of the body tube. It holds the objective lenses and it rotates to enable the magnification. The next part is the arm. The arm supports the upper parts of the microscope and it is used to carry the microscope. This is the base. The base supports the whole microscope and it is used to carry the microscope. The stage supports the slides. Stage clip. The stage clip helps to hold the slide in place. The mirror or the illuminator. It provides light necessary for viewing the specimen and it sends light through the stage opening to the diaphragm. Diaphragm. It is located below the stage opening and it regulates the amount of light that can enter the lenses. The coarse adjustment knob. It raises and lowers the stage objective lenses and used only when focusing the low power objective lens. The fine adjustment knob. It raises and lowers the stage or objective lenses and used when focusing the medium power and the high power. Now let's have a game, the Who Am I? Let's see if you still remember the parts and functions of the compound microscope. Question number 1. It contains the first lens and it is located on the top of the body tube. Who am I? Yes, you are correct. I am the eyepiece. Question number 2. It regulates the amount of light that can enter the lenses. Who am I? Yes, you are correct. I am the diaphragm. Question number 3. It helps to hold the slide in place. Who am I? You are correct. I am the stage clip. Question number 4. It supports the whole microscope. Who am I? You are correct. I am the base. Question number 5. It is located on the nose piece at the bottom of the body tube. Who am I? Yes, you are correct. I am the objective lenses. Great job, everyone! Congratulations!
After knowing the parts and functions of the microscope, this time, I'm going to demonstrate the steps of using the microscope properly. Are you ready? First, place the microscope on a flat, sturdy, and clean surface. Always carry it in an upright position using 2 hands, one in the arm, while the other supports the base. To avoid contaminating the specimen, wash your hands thoroughly. You may also wear proper laboratory gowns, gloves, or goggles depending on the specimen to be studied. Get the specimen you'd like to study and put it on the glass slide. By touching only the edges, place the glass slide on the stage and secure it with the stage clips. Adjust the mirror until it reflects lights upward. In some microscopes, they have light to turn on which serves the same purpose. Adjust the diaphragm until you get the maximum amount of light. Start with the low power objective. Always start with the lowest magnification. Look through the eyepiece and keep both eyes open. Adjust the force adjustment knob until the specimen becomes clearer. Rotate the revolving nosepiece to use the high power objective. You will hear a click sound when the objective lens is in place. You may use the fine adjustment knob to sharpen the focus. This point, never use the force adjustment knob because it will break the glass slide. Finally, when you're done examining the specimen, clean the glass slides and cover slips. Store the microscope in a dust-free cabinet. And now we are going to have a game, and it is called Face Game. I have two faces here, the happy face and the angry face. Now, I will be reading a statement, and all you have to do is to identify if the statement will fall to happy face or angry face. If the statement is correct, I will put this to happy face. If not, to an angry face. Are you ready? Alright, let's start now. First question. Secure the slide with stage clips. Your answer in 5, 4, 3, 2, 1. It's happy face! Very good! Now let's proceed to the second question. Focus the image first with a fine adjustment knob. Is it in a happy or angry face? In 5, 4, 3, 2, 1. You got it right! It's angry face! Let's have the third question. After using the microscope, leave it anywhere. Is it in a happy or an angry face? In 5, 4, 3, 2, 1. You are correct. It's angry face. Let's have the fourth question. You will hear a click when the objective lens is in the correct position. Is it in a happy or an angry face? In 5, 4, 3, 2, 1. Excellent! It's happy face! Let's have the final question, or the fifth question. You may use your shirt in cleaning the lens. Is it in happy or an angry face? In 5, 4, 3, 2, 1. Great! It's angry face!
Hello students! Are you still enjoying our class for today? Okay, at this point, let me test your knowledge. Let's have an activity. All you have to do is to answer true if the statement is correct. Otherwise, false if it is incorrect. Are you ready? Number 1. Mirror is the part of the microscope that captures light that is usually found near the microscope's base. True or false? Yes, the answer is true. Let's proceed with number 2. Base is a flat like this area under the objectives that is used for viewing. True or false? Correct! The answer is false. Number 3. Eyepiece is a part of the microscope individual looks through and makes one see the object. True or false? Yes, the answer is true. Number 4. Fine adjustment knob is a large, round-shaped adjusting knob on the microscope side used to move the upper part of the microscope to focus specimen. True or false? Correct! The answer is false. Number 5. Revolving nose piece is a rotating portion of the microscope under the body tube that holds the objective lenses. True or false? Yes, you are correct. The answer is true. Number 6. Arm is the part where you can hold and carry the microscope. True or false? Yes, the answer is true. For part 2, let's have an activity, I do or I don't. Answer I do if you are going to agree with my statement, and I don't if you will disagree with me. Okay, let's begin. Number 1. Touch the lens with your fingers. I do or I don't. Yes, the answer is I don't. Number 2. Use a high-power objective when using a coarse adjustment knob. I do or I don't. Yes, you are correct. The answer is I don't. Number 3. Carry the microscope using one arm on the base and the other hand on the arm. I do or I don't. Yes, the answer is I do. Number 4. Always start with low power magnifications. I do or I don't. Yes, you are correct. The answer is I do. Number 5. Wash your hands thoroughly to avoid contamination of the specimen. I do or I don't. Yes, the answer is I do. Number 6. Place the microscope on a messy and rough surface. I do or I don't. Yes, the answer is I don't. Job well done, students. Congratulations! To further understand the lesson, I will present a short video clip, and after watching, please answer this question. How important microscope in studying about COVID-19? Enjoy watching!
You might recognize it by now. This is SARS-CoV-2, the virus that causes the novel coronavirus COVID-19. A single cell is just 120 nanometers in diameter. But here it is at 12 million times magnification, a microscopic virus that's turned the world upside down. Each cell of the virus is made up of several parts. On the outside, it's covered in nodules topped with distinctive protein spikes. It's these spikes that give the coronavirus family its name. In Latin, corona means crown, see? And the name COVID-19 simply means coronavirus disease of 2019. The corona protrudes from the glycoprotein membrane, an envelope of lipid molecules that coat the virus cell's body. This fatty layer of skin is very delicate and it falls apart when it meets soap. And that's why, for now, soap is our best weapon against the virus and why you should take your time washing your hands. After 20 seconds, soap penetrates the grease and grime on our hands enough to rip the virus membrane apart, killing it. Under that oily skin is where the real trouble lies. Inside each cell is a small sample of RNA, ribonucleic acid. It carries the virus's genetic code and allows it to multiply, leading to infection. But how does the viral RNA get into our bodies? It comes back to those protein spikes. After the virus has entered the human body, it uses those spikes to attach itself to a healthy cell in the respiratory system. Once that's done, the viral RNA is released and the takeover begins. The cell reads the viral RNA and makes proteins that keep our body's immune systems suppressed. That allows the virus to reproduce and multiply, killing healthy cells and potentially causing lifelong damage or death. So how do we beat this disease? Again, it's all to do with those protein spikes. They might be its strength, but scientists think they might also be its weakness. They now know its genetic code and can recreate it. A trial that began in Seattle is injecting people with that protein. Because it doesn't contain the viral RNA, they believe it's harmless, but it can teach the body how to make antibodies against the protein spikes. Those antibodies would cover the spikes, incapacitating the virus and prevent it from piercing healthy cells. That's the hope. It's just one of about 30 trials underway around the world right now. But most researchers agree a vaccine is still 12 to 18 months away. For now, the best way to avoid becoming infected is to wash your hands, avoid touching your face, and keep practicing physical distancing.
To make sure if you're really grasped the lessons discussed for today, I'm going to give you a test. There are two parts of the test. Part 1 is a multiple choice, and Part 2 is a fill in the blanks. Are you ready to answer the sets of questions? Here it is. And to conclude today's lesson, you're going to answer the assignment. Here it is. In making the slogan, you have to follow the rubrics given.
I hope you guys learned from this video. Stay safe and always be cautious. Thanks for watching. Bye!